Hi, my name is Chris H. and I work with the Acid Integrity Group here at Champion Technologies. Beyond these doors is our H2S lab where we perform testing under sour conditions. Due to the deadly nature of this gas, our technicians perform under full-face respirators and are trained to handle any emergency response situations that may occur. We perform many of the same tests we do under sweet conditions, but here we have the added risk of the H2S gas. This is our bubble test setup, also known throughout the industry as a kettle test, partitioning test, or a green cell test. In this test, we are studying the ability of a chemical to partition from a hydrocarbon phase to the brine phase and onto the metal surface, where we can show the inhibition properties of that chemical. This is our rotating cylinder electrode setup, or RCE. On this test, as well as many others, we run a technique called linear polarization resistance. This allows us to get a real-time corrosion rate inside the cell. These are our RCAs, rotating cylinder autoclaves or rotating cage autoclaves. In this test, we're testing a chemical under high temperature, high pressure, and high shear scenarios. This is our jet impingement loop. Here we have the ability to test chemicals under high temperature, high pressure, and shear stresses of up to 1,000 pascals. We also have the ability to run electrochemical techniques such as linear polarization resistance.
Hi, I'm Glenn Nilsson, Biocide Team Leader with the Flow Assurance Section at Champion Technologies. In addition to providing advanced level field support and researching the phenomenon of microbial influence corrosion, commonly known as MIC, the Biocide team develops Bactron products to provide solutions for controlling bacteria. This particular sample is taken from a gas-producing well. In this particular case, you can see pretty severe MIC which occurred as a result of not properly treating hydraulic fracturing fluid. When dealing with issues of MIC, it's sessile bacteria that are causing problems. Sessile bacteria can be very difficult to eradicate. One of the tools in our arsenal which aids in developing products effective at controlling sessile bacteria is our confocal scanning laser microscope. Confocal scanning laser microscopy provides a level of biofilm studies really second best to none. We're able to build fully direct three-dimensional images of biofilm, rotate them around in space, and even peel back layers and look at the distribution of bacterial cells inside. Here's the biofilm at time zero, and you can see a distribution of live and dead bacterial cells. But the bacterial cells that are dying are starting to fluoresce red, and you can see the red start to overcome the biofilm, at least on the surface. Okay, what's neat here is you can see how he's peeling back the layers of the biofilm, and he can peel them all the way down, then all the way up. And what's particular value, you can see we use a bunch of specialized stains that can differentiate between live and dead bacterial cells. And this aids in biocide development when you're looking at wanting to develop products that will effectively penetrate into a biofilm. And so what you typically see is on the external surface of the biofilm, dead bacterial cells, but then as you peel back the layers, you can start to see, maybe survivors down in the middle, or as you would like to see, you'd like to see dead bacterial cells throughout. So it's pretty cool, real-time video footage of biocides and work. By utilizing advanced technologies such as confocal scanning laser microscopy, it allows us to be able to provide innovative solutions and products to help solve your bacterial issues.